Okay, so I think we'll get started. So welcome everyone to the BMS talk by Ben Gregson. And it'll be on using environmental DNA or eDNA for fungal metabarcoding in soils. My name's Reece Farrer. I'm a senior lecturer at the Centre for Medical Mycology at the University of Exeter, and I'm chairing tonight's talk. During the talk, please do write any questions you have in the chat window, and then at the end of the talk, I'll go through some of those questions, or as many as I can get through. So tonight we're hearing from Ben Gregson. He's a lecturer in ecology and conservation from Anglia Ruskin University. He's a molecular ecologist with diverse research interests, but currently all centered around the use of environmental DNA, paired with next generation DNA sequencing to determine the biodiversity and function within environmental samples. From 2019 to 2020, Ben held a research officer position at the University of Essex. And in this position, he used DNA sequencing to look at the biological and agricultural quality of harvested rainwater collected in water harvesting ponds used by rural smallholder farms in Kenya. And then from 2020 to 2023, he held two senior research officer positions, also at the University of Essex. And in the first position, he used DNA sequencing to look at biodiversity within urban soils compared to natural and agricultural soils. And in the second, he looked at the impact of offshore man-made structures such as oil and gas platforms and wind turbines on the surrounding benthic habitats. All sounds very interesting. So tonight, Ben's talking about environmental DNA for fungal metabarcoding in soils. And take it away when you're ready, Ben.
Thank you very much. Good evening, everyone. Thank you for popping along. So yes, I'm Ben Gregson, a molecular ecologist. So that means I'm using a combination of environmental DNA techniques paired with DNA sequencing. And I use that to answer two questions, basically. Work out the biodiversity, so who is there, and the function, what they can do in a variety of different environmental samples. But this evening, I'm going to focus mainly on soils. So by the end of the talk, you'll work out how I go out to an environment of interest, collect an environmental sample for processing in the lab, how I then process those samples in the lab to actually get the environmental DNA out of it, then how I sequence it, use a DNA sequencer to work out all the orders of the different nucleotide bases, the A, T, C's, and G's, and then use that to make these sort of plots where I can see all the different fungal species present in different environmental samples, in particular soils. So here we've got a variety of different types of soils, and I can see all the different types of fungal species present in it. So I'm going to cover this evening exactly the formal definition of environmental DNA, so eDNA, how we get it out of environmental samples, how we can then use it to work out the biodiversity within that particular sample. I'll go over a few of the projects where I've used eDNA to address different questions, and then give a brief overview of a recently funded project by the BMS, which is again using eDNA techniques. Had a fairly good overview of my career, but I'll just go into a little bit more detail of how I started in this and how I developed all the different skills I learnt. So 2015 to 2020, I did a PhD in environmental microbiology. That was at the University of Essex. Purpose of that was to look at these groups of bacteria that are able to degrade oil. So these are marine bacteria that have the capability to actually essentially eat the oil. And sort of a tag along to that PhD, I got involved in a project that looked at the impact of a offshore oil spill on different microbial communities, so bacteria, fungi, archaea. So here you can see the Agiozoni 2 oil tanker, and this was a oil tanker that hit a rock off the coast of Greece, and then there was a large amount of oil that got spilt in the marine environment, and it contaminated the seawater and the surrounding sediments. So I used eDNA to work out how the microbes were changing in response to that oil, and to try and work out if these oil degrading bacteria were becoming the main dominant players in the environment after the oil spill. Towards the end of my PhD, I had a research officer position. So this was looking at water harvesting ponds based in Kenya. So these are essentially just holes dug in the ground with a little bit of a filter put on it, and they just collect the rainwater. And this is used by rural subsistence farmers in sort of the Laikipia and Mira counties of Kenya. And they essentially use this harvested rainwater to irrigate their crops. And I was interested in sort of the pathogens present, whether these bodies of water acted as reservoirs for different fungal and bacterial pathogens. Following that, I then got, after my PhD, I got 2 postdoctoral positions. One was looking at the impact of urbanization on different microbial communities, again, bacteria, fungi, different invertebrates, to see the impact of urbanization. So as you get closer to a city, how do the microbes compare to a more natural system, say a forest? How do the microbes present in those different environmental samples compare to each other? And then you'll hear a little bit more about that project. This is one of the projects I'm going to show you kind of what the preliminary results were later in this presentation. And then I then looked at the impact of man-made structures on the marine environment. So decommissioned oil and gas platforms and newer installed man-made structures like wind turbines. What impact does the sort of chemicals leached from those structures have on the microbes present in the sediment in the surrounding seawater around those structures? So you can see very, very different angles, very different questions we're trying to address, but all using the same sort of technology to answer those questions. So I'm going to start off with just talk about the importance of fungi. I'm probably preaching to the converted given the audience this evening, but fungi are exceptionally important in shaping terrestrial ecosystems. So what I mean by that is any large landmass that has large amounts of soil and have plants grown within that soil, fungi play a key role in supporting the plant growth within those different environments. So for example, we have different functional groups of fungi that carry out different roles. For example, you have your decomposers. So these break down a variety of different dead material, dead plants, dead animals, waste produced by animals, and then that then contributes to different biogeochemical cycles. So the nitrogen cycle, the phosphorus cycle, the carbon cycle. By breaking down all the organic matter within these dead organisms, then it releases nutrients, and then that makes those nutrients available for plants to grow. Some fungi can also form mutualistic relationships or symbiotic relationships with plants. So for example, they have a give and take relationship. They give each other some sort of benefit. So for an example might be mycorrhizal fungi. So these are fungi that colonize the plant roots in different plants. They help the plant take up more water and nutrients, and in exchange, the plant will provide carbon sources that help the fungi grow. So it's a give and take relationship, and they help each other grow in a particular environment. And then you can also have some fungi act as pathogens, and they're going to be either causing disease or killing particular plant species. For example, Fusarium targets different plants, different agricultural crop species, and they can act as a way to kind of regulate plant populations. So they can make sure that a particular plant doesn't overtake a particular ecosystem. And it's the balance of all these different functional groups that pretty much dictate the ecosystem health, how well the plants grow in a particular ecosystem. So if you have not enough decomposers, then that limits the amount of nutrients available for a plant to grow. If you don't have many symbiotes, then they're not able to, again, plants aren't able to take up as many nutrients from the soil. And if there's too many plant pathogens, then that's again going to limit the ability for certain plants to grow. So if we understand soil fungal biodiversity, this isn't just to answer questions related to ecology, but there's benefits for humankind, because then we can apply some of the knowledge to agricultural systems, and we can use that to increase plant yields, crop yields, and also help us kind of inform how we can conserve all these different natural habitats. Fungi is one of probably the most interesting kingdoms in the tree of life. It's very, very diverse, but the vast majority of fungi we can't actually even see with the naked eye. They're so, so small. So here you have some pictures of some microfungi, some filamentous fungi, and you can see the sort of scale we're talking about, micrometers. So we're talking a thousandth of a millimeter. So these aren't the typical players that you're going to be seeing on your walk through the forest. You might see a handful of different mushrooms, but you're actually surrounded by thousands of spores and parts of fungi that you're not even going to see day to day. And historically, mycologists have obviously paid attention to fungi that produce fruiting bodies, fungi that produce mushrooms and brackets, or whichever fungi might be easier to culture in the lab because you can physically see them or they're very easy to grow. But that's by just focusing on the ones that you can see or the ones you can grow, you're really limiting your full understanding of the fungal biodiversity present in soil or in any sort of environment. How many fungi actually exist within soil? We're talking in the scale of millions. So a lot of fungal ecologists have used DNA-based studies to kind of estimate the predicted number of different fungal species within different soils. And they currently estimate it around to be 3.5 to 5 million different species. Currently, there are only 148 different fungal species described. So these have been, assigned taxonomy. They have a name, they've been fully characterized. We have pictures and we understand sort of the function they play. But we're talking a very small proportion of the total amount of fungi that actually exist within soil. And sometimes you might see these unknown fungi, these invisible fungi referred to as the fungal dark matter. So similar to how we can't detect what makes up the most of our universe, the dark matter, there's loads of different fungi that are not producing fruiting bodies. They're essentially invisible to the naked eye, and we don't really understand much about their evolution, their function, what they are doing, or what particular role they're playing in soil. Typically, if you want to go and do an environmental survey of a soil or of a particular environment like a forest, your go-to method would be a fruiting body survey. So all you're doing is going to your environment of interest. You're recording how often you see a particular fruiting body, so a mushroom, a bracket, etc. And what you might do, you might record the GPS, you might take pictures of the fungi if you can't identify it there and then. You might take sub-samples of it to go culture in the lab or to do some further identification. But one of the limitations with fruiting body surveys is it's highly dependent on when you go out into the field to actually work out what fungi are present in that environment. It's highly seasonal. So if you go in the winter, you might miss some that flourish in the summer. And it's highly weather dependent as well. Field work tends to be fairly weather biased. You don't tend to go when the weather's awful, like you might force yourself, but you try and avoid going and doing a field sample or a field campaign in bad weather. But typically, a lot of mushrooms tend to sprout and grow best after there's been large amounts of rain. There's lots of moisture in the air to kind of help their growth. So if you go and do your field campaign outside the window that's best suited for when that fungi grows, it means you're not going to record that fungi, despite the fact that it is there. It's just not particularly showing its full fruiting body at that point in time. So if you miss the window, the optimal window of a field sample campaign, you're going to miss the fungus, even though it is still in the soil, still growing, it's just not fully showing at that point in time. And there's large groups of fungi that just don't produce any sort of fruiting body at all. So mushrooms obviously produce a lot of different fruiting bodies, but there's whole large groups of fungi that don't produce any form of fruiting body. So you've got tiny microfungi, you've got your yeasts like Saccharomyces, You've got your molds, Aspergillus and Penicillium. None of those will produce any visible fruiting body. And the only way you can properly observe parts of the fungi is, via a particular microscope. Let's say best case scenario, you do pick a fungus that produces a fruiting body. What you're going to need is someone that knows exactly what that fungus is. And, this might be someone that's dedicated years of their life to identify different types of fungi, different types of mushrooms. But there's some that look very, very similar, and there's a chance you might misclassify a particular fungus. So he might say it's this particular species, but it actually isn't, just because they look very, very similar in terms of their color or their shape. And it means fruiting body surveys are only capturing a very, very small part of the entire fungal community. It's limited by time, and it's limited by your ability to identify what that fungus is. You have an alternative to actually going out into the field. So you can go collect a soil sample, put it in a liquid, and then try culturing different fungi in the lab. So you just stick your soil in a liquid onto a different type of plate with some growth media, incubate it over a length of time, and then you'll try and identify the fungi based on the colony it produces. Massive limitation of culturing is that we're using standard sort of techniques, so standard temperatures and standard culture media. You're only going to capture less than 1% of the actual fungi that are actually present in that sample. So, for example, you might grow this plate at 37 degrees, but that means you're going to miss all the fungi that prefer to grow at 38 degrees, 39 degrees, the ones that prefer warmer temperatures. What about the ones that prefer colder temperatures? Are you replicating the exact condition to grow that particular fungus? Have you grown it at the right pH? And also don't forget about the symbiotes. So the mycorrhizal fungi need a plant present to colonize the roots to then actually grow. It's impossible to kind of replicate the proper growth conditions for mycorrhizal fungi within the lab. So you're, again, getting a very, very tiny proportion of the actual fungi present in that soil sample. And you can see, even if you choose to do field-based methods, lab-based methods, or a combination of the two, you're never really fully capturing the fungal biodiversity present in that sample. It's either limited by what you can see or what you can grow. And because of the limitations of these methods, that's why scientists have developed a new technique based around environmental DNA, eDNA, paired with another technique called metabarcoding. So these two techniques combined provide a new way for us to work out what fungi are present in soil. Before I go into what exactly eDNA is, I'm going to start with just DNA on its own. So DNA stands for deoxyribonucleic acid. So it's hereditary material, so it gets passed generation to generation in all organisms. Doesn't matter if you're talking about a fungus, a human, a blue whale. All of us have DNA, and that DNA carries genetic instructions that control how we grow, how we function, how we develop, and how we reproduce. Regardless of what organism you're talking about, that chemical structure of DNA always stays the same. So for DNA, typical structure is this sort of two strands, a double helix shape. You know how you've got computer code is made up of zeros and ones? Well, your genetic code is essentially made up of 4 letters. And these 4 letters represent these 4 chemical bases: adenine, thymine, cytosine, and guanine. So A, T, C, and G. That's what dictates how we grow, how we function, dictates our entire genetic code. And the only rule you have to remember about DNA is that adenine A always pairs with thymine. You can see it there. Adenine and thymine form a base pair. And then cytosine always pairs with guanine. So your C is always with your G. A with T, C always with your G. That's your DNA. While the chemical structure of DNA is the same regardless of what organism you're talking about, what's really important is actually the order of those A, T, C's, and G. So you can see G, T, G, C, A, T. The order of that is very, very important because it dictates the function of whatever the DNA codes for. So you might have a particular gene that's coded by a segment of DNA. Take a fungus, it might have a particular gene that codes for a protein that carries out a function that benefits it. So it might produce an enzyme that helps it break down decaying wood. Like the decomposers have different digestive enzymes that help them break up different materials so that they can acquire nutrients and allow that for growth. If say we swapped around some of these letters, that might not necessarily make the same enzyme that carries out the same function. It might completely change the entire function of the gene that it's coding for. So really, really important that we know the kind of the order of these DNA sequences. Moving on to eDNA. So eDNA, environmental DNA, is essentially a genetic imprint that says a particular species has or is present in a particular environmental sample at a particular point in time. Formal definition is DNA that we obtain directly from an environmental sample. We're talking about soil this evening, but it could be literally any sort of environmental matrix, water, air, sediment, and it's been obtained from the environmental sample rather than directly sampled from an individual. So your sort of classic example, we'll go to water and then we'll come back to soil. Imagine you have a fish that's swimming through a river. The source of eDNA, I would sample the water that a fish has swum in rather than sampling the fish itself. So as the fish is swimming through the river, it's releasing skin cells, it's releasing DNA, microbial cells, some of its waste, its poop, it's some microinvertebrates that are growing on its skin. They might be forming carcasses. So remember, eDNA from the environmental sample rather than the actual organism itself. And soil is absolutely chock full of eDNA because soil is home to a variety of different biodiversity, from your bacteria, archaea, extremophiles like your tardigrades, microinvertebrates, macroinvertebrates, insects, and of course, different types of fungi and plant roots, huge amounts of sources of potential eDNA. And then that eDNA is providing essentially a snapshot of a genetic snapshot of who is present in this soil and what is the biodiversity within that soil. What species are present in this soil here? Next concept to just kind of go over is metabarcoding. So metabarcoding just means we're amplifying and sequencing a particular barcode within the eDNA that tells you what species are there. So for example, I've got a plate here just with loads of different stuff growing on it. I extract the eDNA from it. I then do a few other steps to kind of work out what barcodes are present there. And then based on the order, remember DNA, A, T, C's, and G's, based on the order of those A, T, C's, and G's, I can work out what particular fungal species there are. So this particular DNA sequence would refer to a particular fungi that's present there. For metabarcoding, just imagine when you go into your supermarket, every product has a barcode on it, and that barcode tells you what that item is. It's exactly the same principle. We're essentially using a particular gene, a particular segment of DNA that tells you what that fungal species is. So a barcode in a supermarket tells you what it is, exactly the same. A barcode gene tells you what fungal species is present in your soil sample or any environmental sample. So what is the barcode specifically for fungi. It's a region called the ITS region, which stands for the internal transcribed spacer region. So in fungi, you'll have these other genes, the 18S gene, the 5.8S gene, and the 28S gene. And in between these three genes, you've got two different ITS regions you can target. So it's these little white sections here that represent the barcode. So this short section here will say, I'm this particular fungal species, or this section here will say, I'm this particular fungal species. These three genes, which the ITS regions are in between, are called conserved genes. So it means despite millions of years of evolution, these genes basically have a very, very similar structure to how they did millions of years ago, and they're shared across all different fungi. Whereas these ITS regions are called variable regions, which means they have a very unique DNA sequence, a unique barcode that says, I am this particular fungi. And with metabarcoding, we use something called fungal primers. So a primer is just a very, very short piece of DNA that can bind to a strand of DNA, and it's specific to that ITS gene. So this primer is going to be going across the DNA sequence. It's going to try and find where the ITS region is. Then what we do, we use a technique that I'm going to explain later called PCR to make lots and lots of copies of that gene. Sequence all the amplified genes. So get that order of the A, T, C's and G's. And then that will tell us which fungal species are present in our soil sample. Some of the advantages of eDNA is, obviously we were talking about the microfungi. Using eDNA, we're able to detect microfungi. We're no longer dependent on just seeing things that produce fruiting bodies, or they are observable fungi. You get a much more comprehensive overview of the biodiversity because you're detecting multiple fungal species at the same time. You can also easily collect eDNA all year round. So even if there's seasonal variations between, say, summer and winter, you're not ever going to miss a particular fungal species. And in theory, there's less expertise required because you only need one person to go out and collect your environmental sample. And then for the species identification, you don't need to spend 5, 10, 20 years knowing fungal taxonomy. A computer does all the heavy lifting for you and says, this DNA sequence represents this particular fungal species. So first thing we need to do is actually collect the eDNA. Talk about soil this evening, but I'll tell you how I do it for all the three main environmental matrices. So if you want to collect eDNA from the air, you use this fancy machine called a Coriolis air sampler. All it is essentially a fancy vacuum that sucks in air and dust, and then that dust gets collected with a little watery solution called a buffer. You then extract the eDNA from that buffer rather than the air itself. If you're collecting eDNA from water, what you would do is use a syringe, pass the water through a particular filter called a nitrocellulose filter. That's going to trap the eDNA within the filter. You then break that filter into small pieces and extract eDNA from that. And then soil, you've got two options. You can either scrape soil across the surface, so you're just collecting the top soil, or you can use this metal thing called a soil corer. You'll dig a little hole within the soil and get a long piece of the soil, and then that might represent both the top layer, which is really oxic, has lots of oxygen, and the bit that's anoxic, doesn't have very much oxygen present in it, and you'll have like anaerobic fungi will be growing in this sort of layer of the soil. Then after you've collected your environmental sample, key thing is then you need to store it either at -20, which is your typical freezer, or -80. Otherwise, eDNA will start to degrade due to changes in temperature all the UV from the sun. Okay, so I've gone out to the environment, collected an environmental sample. Now I'm going to take it into the lab and get the eDNA out of the sample. First thing to do is a step called cell lysis. So you introduce a chemical called a detergent, very similar to the stuff you get in your washing up liquid in your kitchen. And what that detergent does, it disrupts the cell membrane of the organism you're interested in. So it would disrupt the fungal cells. By disrupting the membrane, it basically makes the cells burst open and release all the material inside the cell. So importantly, it releases your environmental DNA, but it's also going to release all the other stuff in the cell, proteins, fats, metabolites, everything, absolutely everything that's present within that cell. Then you're going to introduce a few different chemicals. You're going to do a step called inhibitor removal. You can use a variety of different chemicals to get anything that is not related to eDNA out of the liquid. In particular, with soil, you really want to remove these chemical compounds called humic acids. So when we get to the later steps, some of those steps can be impacted if there's too much humic acid present in your extract. So key thing is introduce a load of different chemicals to remove all the other gunk that isn't related to the eDNA. Next step, we do something called a spin column-based nucleic acid purification. For this, you just have a little tube, a little spin tube, and within this white section here, you have a little membrane that's going to trap eDNA. So the actual membrane is made of silica. You then introduce a little chemical to your extract that makes your eDNA very, very salty and covered in sodium ions. This then means when you have salty eDNA, it's then actually going to bind to this membrane here. So you're only going to be trapping eDNA rather than all the other stuff that's been released from the cell. So once you've added your really salty solution, you put your eDNA in here. You're then going to spin that tube in a big centrifuge that's going to spin the tube round thousands of times a minute. And by spinning it, it draws all the liquid through the tube. The liquid collects at the bottom. So you've got loads of waste here at the bottom of the tube, but all your eDNA is now stuck in this little white membrane here. Once your eDNA is bound to that membrane, you then just add some ethanol. So, the stuff that's in your hand sanitizer. Use some ethanol to just get rid of anything else that is not environmental DNA. You're going to remove salts, more of those humic substances, those chemical compounds we want to get rid of. Anything that's not related to the eDNA, add ethanol to it. Again, spin it down a couple of times, get rid of all the ethanol in the tube. Then your final step is the elution step. So here you just add another chemical, a tris buffer, or maybe something like water, and that's going to release the eDNA from that silica membrane. And after all that work, typically what you're left with is a tiny, tiny tube, a little bit of liquid left in the bottom. And this is usually 50 to 100 microliters of eDNA. So microliters, you're talking 1,000th of a milliliter, very, very small volumes at the end of it. Okay, we've got your eDNA out of your sample. Now what we want to do is make lots and lots of copies of that barcode. So remember, we want to do metabarcoding. So we want to get lots of the gene that tells us what the fungal species are present in that sample. And to do that, we do something called PCR. I think a lot of us are more familiar with PCR since the COVID pandemic because, you do your swab and then you get it sent off for a PCR test. I'm going to go over the steps of how a PCR works. So idea is you're going to make millions of copies of that barcode gene. So you've got your DNA here at the top. First step is called denaturation. All you're doing is just heating your DNA so that this double strand ends up becoming two separate single strands. Next step is annealing. You lower the temperature so that it then allows those fungal primers to bind to that ITS region, binds to the particular barcode that we're interested in, raise the temperature again, and then do a step called extension. So it's going to get individual bases and make sure that there's pairing. So if this was an A in your DNA, it would then bring a T across, and then you'd have a complementary base pair there. If there was a C here, then it would pair with a G. And at the end of it, you have two identical genes of interest, and then you'd run the cycle again. So you start with one strand of DNA, then you've ended up with 2 copies of the gene that you're interested in, then you'd repeat, so you'd have 4 copies, 8 copies. By the end of it, you've got millions of copies of that barcode gene. And this is just showing again how the PCR process works. Remember, it's the ITS gene is the one that we're targeting for fungi. But if you're interested in other species, then you would target different genes. So if you're interested just in fish, you target the 12S barcode. If you're interested in a variety of different eukaryotes, so different metazoans, fish, shells, invertebrates, coral, you might target the CO1 gene, CO1 barcode. Similar for the 18S gene, again targeting more groups of eukaryotes. Or if you're just interested in, say, bacteria, archaea, you target the 16S gene. So different genes you target based on the group that you're interested in working out what is there in your sample. We're interested in fungi, so we're targeting that ITS gene. Now I've been able to get the eDNA out of the sample, amplify up the ITS gene. Next thing I need to do is work out the order of those A, T, Cs, and Gs. So you can see we've got some DNA sequences here, and it'd be G, T, A, G, G, A, A, but we want to do that for all the sequences that are present in our environmental sample. And the way they used to do this back in the 70s, they do it manually with these special gels, and they'd read the gels and go, that's an A, that's a T, that's a C, that's a G. Obviously, that was very, very, very slow. So they then developed these automated DNA sequencers. So here you've got the PRISM DNA sequencer that was developed in the 1980s. But now there's these newer next generation sequencers produced by companies like Illumina and Oxford Nanopore that could produce much more data, millions and millions of DNA sequences in a very short period of time. The way I think about DNA sequencing, I kind of use this analogy to teach some of my students. Imagine this text represents all the eDNA you've just extracted from the soil. A PCR primer, those short pieces of DNA that are targeting the ITS region, are basically kind of the Control F, Command F function. So if I'm looking for a particular word, I'm looking for a particular region of the eDNA that the primer's going to bind to. And then the PCR is just amplifying all the stuff around those found words. So these highlighted words represent the entire barcode that needs to be DNA sequenced, that I need to work out the order of the letters to work out what fungal species are present in the soil. Now, as humans, we've got no easy way to actually read that text. So what we tend to then use are these automated DNA sequencers. Here you have an Illumina NextSeq. So this is one of the most recent models of Illumina DNA sequencer. Very, very expensive to buy, roughly around £200,000 to buy. And then every time you run it, it costs around £4,000. But usually you're running lots and lots of samples at the same time. So sounds expensive, but usually it's relatively cost effective. It's just very expensive to get the DNA sequencer in the first place. Illumina is kind of the key, the leader in the market in terms of DNA sequencing. The way their DNA sequencers work is these little strands here represent the 2 individual strands of DNA. What they'll then have, so say this red one represents an A, you'll then have a T bind it. So remember, A to T, C to G. And when that base binds there, it releases a fluorescent light, a particular green light. So here we're saying green represents T. This orange one might represent a C or a G. You essentially have a camera at the top of the DNA sequencer that's watching all these color changes hundreds of thousands of times simultaneously. And from that, it's then able to work out the order of the A, T, C's, and G's. Just individually, the first cycle, here's your T coming that's green. Next one's a red, so that's an A. This one's an orange, so this could be a C. You've got another red. So you can see, based on the colors, it tells you whether it's an A, T, C, or G. A more recent DNA sequencer, this really fantastic technology called a MinION, is developed by a company called Oxford Nanopore. USP of this, it's the first portable DNA sequencer that in theory you can take anywhere in the world and you can hold it in the palm of your hand. It doesn't require Wi-Fi, so here you can see it being used in Antarctica to kind of measure antibiotic resistance genes present in Antarctica. Very, very portable, very, very cheap as well. Remember the other one's about 200K. This one you can buy along with a chip for about $1,000. And the technology is very, very different. Rather than producing very, very short amounts of DNA, reading 150 to 200 base pairs, it actually can read up to 2 million. So you can get entire strands of DNA read at the same time. This is how it works. So actually on the chip that comes with the MinION DNA sequencer, there's these tiny, tiny holes called nanopores. At the top of the nanopore is a little enzyme that just unwinds the DNA for you, and then a single strand of the DNA goes through the hole. There's an electric current going through the DNA sequencer, and as the DNA passes through this little hole, there'll be a change in the electrical signal. So if it's an A, it would cause a change in the signal that's distinct, versus if it was a T, then it would change the electric current in a different way. So A, T, C, and G are read based on how much the DNA disrupts the electric signal passing through the DNA sequencer. And then you've got an argument, which one's kind of the best one to use? Should I use Illumina or should I use the Oxford Nanopore sequencer? And that probably depends on the question you're trying to answer. So as I said, the Illumina DNA sequencers only really do very, very short pieces of the DNA, 150 to 300. So you only get a very short amount of the DNA actually sequenced. So you get a much smaller piece of the puzzle filled. And that's good if you're just targeting these variable regions. So if we're just targeting a small segment of that ITS gene, then that's great. We can answer the questions. The nanopore DNA sequencer fills in much larger pieces of the puzzle because we can do up to 2 million base pairs. So if you remember what the ITS region looks like, you could also sequence the other genes around it. You could do the 18S, the 5.8S, the 23S, and both of the ITS regions in one go. And then with that, you have a much more, with that data, you're able to assign taxonomy and say what that fungal species is with a lot more confidence. But it's weighing it up because these portable DNA sequencers don't produce as much data as the Illumina sequencers. So it's weighing up more data versus more confidence in your data. Got the DNA sequencing data off the sequencer. Now what we have to do is compare the A, T, C's, and G's to a big database that has, for all known species of a particular organism, it has the DNA sequence that says, this is the sequence for a particular fungi, and you'll compare the DNA sequence that you've got off the sequencer with what's in the database. So for fungi, we have a DNA database called Unite, and this has a load of different ITS DNA sequences that says this is a particular species of, say, Fusarium, and this is another particular species for a different, this is another DNA sequence for another type of fungal species. What you've got off the DNA sequencer, say we just got this random selection of letters, we'd compare it to what's present in the database, and then we can basically say this is the particular fungal species based on how similar it is to what is in the database. And after all this work, this would be one of the outputs that we'd get from that data. So the y-axis would show you the number of fungal species present in a particular sample, and the x-axis would say a particular group you're interested in. So this is from the project that was looking at urbanization on soil communities. So we've got agricultural soil from farms, some from green spaces like parks, industrial areas. Legacy represents an area that had a particular function before it became an urban area. Natural soil like a forest, and then residential areas, places with lots of flats or lots of people. And what we can see is, interestingly, the number of fungal species that we collected across the UK. So we've collected a variety of different agricultural soils, urban soils, and natural soils. We can see agricultural soils and natural soils have, on average, a lower number of fungal species compared to urban soils. So there's more types of fungi present in urban areas versus natural areas. But that doesn't necessarily mean they're good fungi or bad fungi. Do we know whether they actually support the ecosystem services provided by the soil in any way? Are they pathogens? Are they decomposers? Symbionts? We have to work that out. So another thing that we can get from the DNA sequencing data is the actual fungal community, which fungi are actually present there. And interestingly, from the same data, we could see there were way more plant pathogens, so Fusarium, Pseudopithomyces. Here you can see a little stacked bar plot. The y-axis adds up to 1. 1 represents 100% of the community. So here you can see we're talking up to about 60% of all the fungal species present. And you see a huge chunk. So Fusarium, represented by yellow, the yellow bars. So we can see lots and lots of Fusarium in all the urban soils, but in the natural soils, there's barely any. Same for the Pseudopithomyces in the light blue, more again in sort of the urban soils compared to the natural soils. Also, interestingly, remember the mycorrhizal fungi, the ones that help the plants grow by colonizing their roots. Clavunilopsis, very, very present in natural soils, but completely absent in all the urban and agricultural soils. So you sort of take-home message from that is urban soils aren't particularly a great environment for plants to grow, which kind of makes sense when you look at the plant species supported in a big city versus a forest. So there might be more fungal species in urban soils, but it looks like the actual types of species that are present aren't going to be particularly helpful for supporting plant growth. Now, I've obviously I feel like I'm trying to sell you eDNA as this, it can solve all problems, but there are some challenges when you work with eDNA. Very susceptible to contamination. Sometimes you have to work with a full bodysuit, proper gloves, disinfect everything, and minimize eDNA from another sample contaminating the sample you're working on. So if you have a few spores from another sample get into your soil, then you're going to misrepresent what's actually present in the soil. Spatial heterogeneity just means in a very short space, even, like I could collect 2 soil samples a meter apart, and the actual fungi present could be very, very different. eDNA is just a snapshot. So just because I detect eDNA from a fungus is it still present in the environment, or was it present in the environment, say, a few months ago, maybe a year ago? Is it actually biologically active, or is it just dead and not actually contributing anything to the ecosystem? You also have that a lot of those databases I talked about aren't complete. So a lot of the eDNA will just come out as unclassified, a novel fungal species, but I can't tell you what it's closest to. And one of the key issues with particular fungal eDNA is we don't know which region to target. So remember, there's two different ITS regions we can target, either the ITS1 region or the ITS2 region. And we can design primers that target either one or the other. And there's pros and cons depending on what question you're trying to answer. So I might target ITS1 because it's able to pick up more fungal species. But however, when you amplify up that gene with the PCR, the actual length of the gene really varies. So sometimes it could be really, really, really short or quite long. If it's really, really short, then I can't actually say what the fungal species is with very much confidence. But ITS picks up not as many fungal species, but the length is very consistent. So I might miss particular fungi functional groups if I pick ITS2 over ITS1. And basically, there's no consensus on what is the best region to target. So I was recently awarded a research grant by the BMS to kind of try and answer this question. We know that whichever primer you choose, you're going to have some sort of, depending on which primer you're choosing, you're either going to miss particular species, and we don't know which is the best primer to pick depending on a particular soil type. So if I collected a tropical soil, the soil is very highly degraded and there's not very many nutrients in it, and that's going to influence the fungal communities in it. And I don't know whether it's better for me to pick the ITS1 region or the ITS2 region. So I want to look at loads of different soil types over different climates and try and work out which is the best ITS region for me to target, ITS1 or ITS2. So what we're going to do, we're going to collect eDNA from 9 different soil types. So there's going to be 3 types of land uses: agricultural soils, so from farms; natural soils from places like forest and grassland; and urban soils from built up cities and industrial parks. We're also going to try and cover all the different climates. So you're going to have some from tropical areas, temperate areas like the UK and Europe, and then much colder areas. And we're trying to get a really global overview of fungal biodiversity. So we've collected samples from Austria, Indonesia, Kenya, New Zealand, Norway, and the UK. And we're going to test different primer sets. So we're going to try 2 different primers that target the ITS1 region, 2 primers that target the ITS2 region, then 1 that targets the 18S gene, because you can use that as an alternative marker. And you can see the variety of different soils we're getting, some from those rural subsistent farms in Kenya, some urban soils collected just outside the old Olympic Stadium, and then some collected from Svalbard, so in between Norway and the North Pole, these little natural grasslands present there, a huge variety of different soils. We're going to try and collect and work out what's the best ITS region to target for that particular sample. And the impact of that will hopefully be that we're going to provide a guide, not just for researchers like myself, but also for these environmental consultancies and environmental companies that are starting to come out and offer eDNA as a service, essentially. So the market leader currently is a company called Nature and Metrics, but you have other companies, Applied Genomic, Crestwood Environmental, and government agencies like the Environment Agency that are starting to use eDNA to monitor different environments. And hopefully we'll have a guide that says, if you collect, say, a tropical soil, we recommend you use this particular primer set that targets this particular gene. And that's going to give you the most comprehensive overview of the species present in that soil. And by doing that, we then support better reproducibility. Another limitation of eDNA that sometimes the methods aren't standardized. So if we have everyone using the correct and same primers, we're going to get similar results that are comparable. And hopefully from that, we'll also publish these results in a journal like Fungal Ecology. And hopefully I can present some of the results at a later date. So in conclusion, I hope I've showed you the power of eDNA, how it's essentially revolutionized fungal ecology. However, there are limitations to eDNA. We need to make sure that all the methods are standardized so we have a much better understanding of all the fungi present, all this amazing life beneath our feet that we don't fully understand to this date. So thank you all very much for listening. I hope you've enjoyed the talk and welcome any questions.
Great talk, Ben. Thank you very much. Very comprehensive. There's lots of questions that have come through, so
I will just stop sharing. There we go.
great. So we'll go through them one at a time, but maybe first I'll quickly ask you a question. So how confident are you with this kind of barcoding approach, perhaps in comparison with something more like metagenomics, where instead of taking a barcode of just the ITS, that you'd look at potentially the whole genome?
I think metagenomics is a lot more informative versus metabarcoding because you don't just get taxonomy, you get information in terms of function. So you can not just get this is this particular fungal species, but this is also what they can do, what they can potentially do. I think metagenomics is probably going to overtake metabarcoding as a method. Some of the limitations of that are obviously the price, probably what, 10 times more expensive versus metabarcoding. And also in terms specifically for fungi, a lot of the softwares has mainly been designed more around bacteria. So I think there needs to be more development that target specifically fungi, but just eukaryotes generally. And also, I think a lot of the software has been developed more with a medical focus in mind. Interested in stuff around the gut or bacterial pathogens. So there needs to be a little bit more development in the databases used for the taxonomy assignment as well.
Nice. So the first question was, how limited is metabarcoding given so much fungal diversity is uncharacterized? And is metabarcoding different from amplicon sequencing?
No, they're essentially the two terms that mean the same thing. So the amplicon that I would be targeting would be the ITS gene. You see them used kind of interchangeably. You'll also see them, some people call it metagenomics as well, when it's not really metagenomics. It's more a form of metagenetics because you're only targeting one gene. And yeah, I did highlight it in my talk that metabarcoding is hugely, hugely limited by the databases available to you. They've done a huge amount of work building up the Unite database, but there's loads and loads of issues with it. Particularly, I mean, we're just talking about terrestrial fungi. If you're really interested in marine fungi, a lot of that database is biased towards terrestrial ecosystems versus marine. So yeah, there are huge limitations, and the confidence of our data is highly dependent on the completion of the fungi. And realistically, we're never going to have a fully complete database available.
Yeah. So earlier on in your talk, you were giving details about your library preparation and DNA extraction. So there's a few questions about that, including what can be the impact if you keep your soil sample at room temperature before eDNA extraction?
Realistically, then you might have changes within the microbial community. So you'll have, say, particular groups that prefer to be, it's dependent on where you've collected the sample from and the difference between the temperature present in that environment and the room temperature of your lab. But you might get changes within, in a very short space of time, changes from, say, if you've collected it from a really cold environment like the Antarctic and the Arctic, you have a transition from your psychrophiles, so microbes that prefer to grow at much lower temperatures, to more mesophiles, ones that prefer to grow at the slightly warmer temperatures. So you have a transition in
Mm.
the microbial community, and it means when you sequence it, it's not really going to be representative of what is properly present within that sample when you collected it. So that's why the preservation is very, very key, the -80 or the -20, just to cryopreserve that snapshot in time, and then ensure that straight after you've taken it out of the freezer, directly go to the extraction.
The next question was, do you know how accurate the fungi barcode database is? I think you spoke about that a bit, including Unite, was it? And then taxonomic expertise must be vital at the stage of compiling that. Also, is there one central database that people use, or are there different ones?
So Unite is the main database used for fungi, and it is also highly dependent on which marker you're using. So if I'm interested in the 16S gene, which is mainly used for bacteria and archaea, I use a completely different database than Unite. Unite is fungal specific, so I'd use green genes or silver if I was interested in bacteria. If I'm interested in more a variety of different eukaryotes, then I might use the Midori database for the kind of the CO1 genes. If I'm interested in protists, I use the PR2, which targets the 18S gene. And I completely agree that you still need the expertise to inform those databases. They have to be complementary as well. And usually your results you might have to then confirm with maybe some culture-based techniques on top. So you might have culture-independent techniques, your eDNA, your sequencing, and then you'll pair that with then trying to confirm the presence of that fungi or organism in a different way.
Hmm. Okay, this seems like a tricky one, but how would you know if a specific fungi or fungal group is culturable at all if it's not already in the literature?
So this is probably where the metagenomics will actually come into play. So metabarcoding is probably not the best technique if we want to start culturing the unculturable.
Hmm.
So with the metagenomics, we're going to pull out whole genomes, so metagenome assembled genomes. And from that, we can then kind of work out the growth preferences of the
Hmm.
fungi. We might see particular genes that show it grows in a particular way, or it might have a preference to a particular temperature, pH, and then we can make adjustments for that. There's huge developments in trying to culture the unculturable that's been informed by metagenomics and metatranscriptomics.
The question was, can metabarcoding identify at the species level or only at the genus level?
So that is dependent on the technology you use. So if you use Illumina sequencing, so that really short thing that's either 150 to 300 base pairs, you can realistically only go down with confidence to the genus level. Whereas if you use the Oxford Nanopore and you get a really comprehensive length of the gene, then you can, at that stage, say with a lot more confidence and assign to the species level.
Hmm. Interesting. Um, there's a question possibly regarding library prep still, asking what's the best way to remove inhibitor?
That's kind of highly dependent on the kit, huge amount of different kits. So typically, I tend to use the Kaigen Power Soil kit, and that seems to have never really failed me. I'm not sponsored by Kaigen, but I'm just saying that it's never really failed me, and the chemicals it uses there to remove the inhibitors never really had any sort of issues. And I've collected soils from really extreme environments and never had an issue with inhibition really. Sometimes, obviously, with every PCR amplification, you need some sort of optimization, but it's never really been a huge issue for me.
You were asked that your approach is going to shift the bias away from the dye carrier, the Basidiomycetes and Ascomycetes, in sampling and subsequent data sets. What domains do you expect to have an explosion of data given this new method?
So, which domains in terms of
I guess fungal, is there some undersampled phyla that you'd expect to, I mean, have you seen from your data that it's any unexpected groups that are more present than expected? I don't know.
um, I mean, do you mean in terms of when I pick ITS1 and ITS2, or
I
just
think this is, I think the question is outside of the dicarria, are you finding many fungi?
in the sort of the data sets I've collected, not a huge amount of variety outside those groups, no. But again, that again is likely due to bias within the databases.
Okay.
I always report how many is unclassified within any paper that I publish, just to make, be open and transparent about how much of the data has been unassigned to a particular group. So
Typically, what percent would that be?
highly sample dependent.
Okay.
Anything, the most I've seen is 80%, which is
Okay, quite a lot.
quite a high, and then lowest is probably single digit percent, I would say.
Okay, that's interesting. Are there certain locations that have a higher percent of unknown samples
And
typically, or
usually very extreme environments. So ones from Antarctica, a very high level that with no confidence. So which makes sense.
yeah, interesting.
So you see that
Um,
basically by how easy it is to collect a particular sample is going to bias all the databases as well.
so there's a question here saying, can eDNA be used in conjunction with fruiting body surveys to track spore transport outside of normal ranges, e.g., as climate change and anthropogenic activity forces animals into new ranges, their fur, etc., will carry spores into new places?
Yeah, I absolutely think you could use this approach. Probably if we're interested in spores, then the air sampling via the Coriolis would make the most sense in terms of the sample collection. And yeah, absolutely need to do complementary approaches. If there is the funds and resources available to do multiple things to validate an eDNA data set, then you should do it. So fruit in body surveys on top of additional analyses would be perfect, and would be preferable.
There's a question about how you cope with sparse and noisy DNA databases. Is there any way of dealing with that?
Uh, in ter- Not really. You just have to kind of work with what you're given. I think that's
Yeah.
you mantra.
Um, you asked, how do you decide how many samples to take to give you an accurate measurement of diversity, i.e., how far apart are your cores, and does this vary according to time of year and fungal growth rates?
So typically I will do within say I'm doing a forest, I would tend to do three transects, and then within that there'll be 10 quadrats, and then within that quadrat I'll do three cores that are just a few centimeters apart from each other, just to make sure, assess the spatial heterogeneity essentially. So try and get a really comprehensive overview of the environment. And the more samples, the better. So if I see a number and I can add a zero at the end of it to really get, enhance the confidence of my data, then I will do that.
Can you explain the low concentration of DNA in soil that you found? And when you're doing the DNA extraction?
Did you mean in terms of the volume at the end of it?
I think so.
Essentially, the reason we use such a small volume of liquid is the more liquid I could add multiple mil if I wanted to, but then you have the chance that you're going to dilute your DNA to extinction, where you can't actually either amplify it up or you just completely wash it away entirely. So 50 to 100 is pretty much regardless of which DNA kit you use. That's probably the bog standard that you would use. If you really want to make sure that it's all come
Hmm.
off the filter, you can use a higher volume, but it's always weighing up that you don't dilute the DNA too much. So yeah, that's the reason for 50 to 100.
And how important is homogenization in eDNA processing, and what are the best ways to prevent eDNA degradation?
So homogenization, very, very important. Usually, along with the chemical lysis I mentioned in my presentation, there'll be some kind of mechanical lysis as well. So this might be essentially the tube will be shaken at very, very high speeds for a short length of time, or it will be shaken at a medium speed for a longer length of time, say 30 minutes to an hour.
Hmm.
Uh, in terms of
Fine.
in terms of eDNA degradation, you just have to essentially work. We have our own specific lab designed for eDNA work. Low light, ensure there's no UV radiation, so no sunlight coming into your lab because it's very susceptible to UV degradation. Absolutely sterilize everything in ethanol, bleach, because you can have microbial degradation of eDNA. And then just ensure no AC, stable temperature, very, very susceptible to degradation if there's large shifts in change up or below.
Interesting. Um, you were asked, are there issues with taking eDNA from soil compared to air and water where there is more flow? I can imagine that you end up with a sample which is only representative of a small region of soil.
Yes, but then it's weighing it up. So I would say that is true. But if I'm collecting eDNA from, say, a river, a moving ecosystem, it's very difficult to gauge because if there are not many variations within the temperature and it's fairly protected from UV radiation, eDNA can be quite stable. So just because I've collected eDNA at one point in a river, how far has that eDNA traveled? It's really difficult to kind of work out where that's come from. Like if it's part of, have I been lucky and it is right here, right now, or has that come from several miles down the river Cam or something? So
Hmm.
with eDNA from soil, it's in one area very unlikely to move if it's the lower layers of the soil. So you can say with a lot more confidence that it is there or was there. Again, whether it's biologically active and playing some kind of role, that's a little bit more difficult to disentangle, but easier to work with soil, I would say, in terms of sample collection.
You were asked whether this could potentially provide a new epidemiological approach to tracking the spread and burden of fungal pathogens, for example, chytrid infection with frogs. I suppose if you were doing water, perhaps.
Yeah, absolutely. So there's two different modes of using eDNA. One is to look at the whole community as a whole, or you can just target individual species. So a lot of companies offer sort of crayfish eDNA detection if they're interested in crayfish, invasive species in freshwater ecosystems. You can use it for seahorses. You can use it for protected species like the greater crested newt. So if you're interested in particular critids that are targeting amphibians and they have a huge impact and it caused a huge amount of amphibian decline along with climate change and everything else. But yeah, you can absolutely use this to target a particular species. And you could use a slightly different technique. So you could do a qPCR, a quantitative PCR. So you use primers that are specific for that a particular cryptid fungi that you're interested in, and you're just going to do the PCR reaction similar as you did as I explained in my presentation, but you do it in the presence of a fluorescent dye, and that can tell you basically the presence or absence of that particular fungal species and roughly how much of it there is.
Hmm.
And I would usually do a qPCR sometimes as a validation for a particular group, depending on the research question I'm trying to answer.
Yeah, I can imagine a situation where there's perhaps certain species of high interest that pop out of the analysis, and then you'd want to
And you would just want to confirm.
go back and see if you can
Yeah,
find any evidence.
if you've got the resources, go back and try and get a proper confirmation it's there, or do some additional tests, molecular tests to kind of enhance the confidence.
Yeah. Okay, you've got a longer question here that looks like it's in four parts.
Oh,
So
excellent.
the first one is a person saying that they're familiar with Sanger sequencing, and could you briefly explain what type of sequencing is required? So I think you've kind of maybe gone over that already with Illumina and Nanopore. I don't know if you wanted to add anything more.
Yeah, so Sanger sequencing was sort of when I was talking about the ones in the 1970s and the Applied Biosystems PRISM DNA sequencer that uses Sanger sequencing, and Sanger sequencing is still available. And usually, if you do Sanger sequencing, the taxonomy is you can say with a lot more confidence what a particular species is, but it's not particularly high throughput. So you can only do it either for, say, individual species rather than a community as a whole. And it absolutely plays its role. Again, that could be a complementary approach. If I say culture a fungi and I want to kind of identify what the fungi is, I would maybe extract from that colony and then send it off for Sanger sequencing.
Um, Usman also wondered, I guess, in relation, is saying with Sanger, they receive ABI files containing sequence chromatograms, but what type of files are generated with the type of sequencing you're using?
Essentially fast Q files. So these are just basically the A, T, C's, and G's, along with something called a quality score, which basically says how confident am I that this A is actually an A rather than a T, C, or G. And then that's what we use to analyze the data. And what we're trying to do is pull out particular DNA sequences, so a particular taxonomic unit called an amplicon sequence variant, and then that is a unique DNA sequence that you can then say how often do I detect that DNA sequence in a particular sample.
You've been asked what kind of softwares do you typically use, and briefly explain the processing steps. So I guess taking those FASTQ files, what do you typically?
So I tend to read my data into R using the R programming language. Very common pipeline to use is called DADA2. So that's just the way. So I remember I was describing those amplicon sequence variants. That's essentially how we can pull those out from all of the A, T, C's, and G's. Assigns based on the quality scores and counts all the ASVs in all the different samples, basically. And then for plotting, sort of the community analysis that you saw, then I would usually take the data from Data2 and import it into another piece of software called phyloseq, and then that can do all the sort of phylogenetic community analysis, show you how much of a particular fungi is present in the sample and everything.
And lastly, based on the sequence data, can we validly describe specimens as new to science or as new records, similar to how we do with data obtained through Sanger sequencing?
There usually have to be some kind of additional validation for it, but if you can do some additional work to then, like I say, potentially culture it, then you can absolutely use it to describe new species. You can just say supporting data provided by this DNA sequencer.
You've been asked, is there a DNA extraction method that you can share with us?
As I said, I'm mainly for soil. I use the Power Soil Pro kits. They never done me wrong in terms of soil extraction, and the process behind that I described in the presentation is pretty much fairly similar. Like you might use slightly different chemicals, but to be honest, all the kits are fairly similar. And there's plenty of benchmarking studies that also say maybe for this particular soil, you might want to, this environmental matrix, you might want to use this kit over this kit, but there's very little difference between.
So does the eDNA only include DNA from living organisms, or does it pick up dead material too? And is there any way to tell the difference in your analysis?
Essentially, no. And then this is one of the downsides of eDNA analysis. We do not know when we detect it whether that signal is from something that is participating, that biologically active and participating in that ecosystem. Maybe something to kind of support that would be to do maybe some environmental RNA analysis. So that's rather than, that might show a functional organism, but it's not. It is, as I said, kind of one of the limitations of eDNA work.
There's a question wondering whether you use any different primers to ITS to target even more specific types of fungi or even functional traits. Um, I don't know, have you
Yeah, I
tried
mean,
that?
absolutely, you can use specific primers to target, say, a muscular mycorrhizal fungi. It's highly dependent on the research question you're trying to address. ITS is pretty much the go-to for just, the comprehensive overview. But obviously, if you're interested in particular functional groups, you want to say you want to see the impact of, you're a farmer and you want to see the impact of your new farming practice on the mycorrhizal fungi, then you can absolutely use primers specific to that, sequence it as before, and you can then use that to do exactly the same steps.
Mhm. Another question is, do we need to make a control sample for extracting the eDNA? But how are we setting for sampling from the natural source, for example, Antarctic soil? I'm not sure I fully understand that.
In terms of the control, if you have no control, then your experiment's out of control. So for every extraction I do a blank extraction. So just to ensure there's no contamination in any of the reagents in the kit, and then when I do every time I run a PCR, there's a no negative template control just to ensure, again, no contamination with any of the reagents I'm using. Use a
Mhm.
sequencing control as well. So controls are essential for eDNA work, any sort of molecular work, but absolutely you have to use controls.
I seem to remember a few years ago there was a issue with perhaps some of the purified water or something that was being used that had contaminants in it, and I think that had fallen into a bunch of downstream analysis.
Yeah, essentially what I do also when I'm using my doing my analysis in PhyloSeq, I use another package that can remove. Because I've sequenced all these negative controls, it can then remove any sequences that it sees as similar to the negative control. So it can say this might be a potential contaminant from that was present in because it's present in your negative control. I will remove it from your actual samples.
I'm going to select a bit more strategically as we're beginning to run out of time. You've been asked about ITS 3 and 4, whether those are used. And also someone else, in fact, has asked, would you use 18S?
That we are, it can be used as an alternative marker, and that's why in the BMS funded study, that's kind of acting as an alternative marker. Just in terms of the literature, they seem to be the most widely used regions to target. You can absolutely target ITS3 and ITS4. So the rationale in the BMS project was also they tend to be the regions that companies offer in terms of when they do the library prep and when they do the eDNA monitoring. They'll say, do you want us to target ITS1 or ITS2? So when you've got these different primer sets, I've essentially gone for the two for ITS1. One is basically, one set is the most cited in terms of the academic research, and one is the most, is a commercially available one as well. It's the same principle for ITS2 as well. If I
Let's
had the choice, I would sequence all of them simultaneously, but you always have to, it's an issue of cost as well. If you have a client come to these environmental consultancies and say, I want, I've got a limited budget, then you want to be able to say with some confidence, well, for your particular environment, we recommend based on this study or this report to target this particular gene.
That makes sense. Um, I suppose a good one to potentially end with would be if you had the opportunity to fund a fungi research program, would you focus on cultivation and identification of species or their analysis using eDNA? I don't know, is there a particular branch of topics you'd be most interested in?
In terms of which ones I would target, or
I think it's an open question to which if you could fund a fungi research program, what that would be on.
I would probably use it as sort of a look for particular fungi that produce very novel biocompounds and use those as biofoundries for new antibiotics. So one of my research areas now is using eDNA to work out about antimicrobial resistance
Hmm.
in the environment. And the thought of just our typical antibiotics no longer working scares the hell out of me. So we need to find novel species that produce novel compounds that we could then potentially down the line be used to develop new antibiotics. And so
Yeah.
marine fungi seem to house quite a few different novel classes, potential novel classes of antibiotics that are fairly effective against, say, MRSA or vancomycin resistant bacteria. So that's something I would hopefully try and fund.
That sounds like a very good answer and a very interesting topic to follow up on. Okay, so I think we're running out of time. We're about at the latest, but there are some other questions in the chat window that I assume can be sent to you
Yeah,
to see or
absolutely.
answer afterwards.
I'm happy to answer all of those. So
Okay, so in that case, thank you very much, Ben, and thank you everyone for participating.
thank you very much, everyone.